This is our Capstone project on pharmaceutical chemistry. Our presentation is on Jason Sello's work on the rational design of selective and bioactive inhibitors of the Mycobacterium tuberculosis proteasome. Jason Sello earned a bachelor's degree in biology in 1997 from Morehouse College. From his interest in viruses, he realized that the knowledge he gained could also work to find solutions and cures. During his time at Morehouse, he found an interest in the research of Professor James Hogle at Harvard University. Hogle was studying how viruses invade human cells, and this sparked interest in Sello. After reaching out to the professor, he was able to receive the opportunity to work in a research lab in Harvard through a program called Leadership Alliance. This fostered a love for research and Harvard, as he went on to receive a PhD in biophysics in 2002 at Harvard University. After that, Sello became a postdoctoral fellow in Harvard Medical School, studying enzymology of chloramphenicol biosynthesis. He became a professor at Brown University and is currently a professor at the University of California, San Francisco, in the pharmaceutical chemistry department. Sello believes that all students should be given the opportunity to be in research and that the field requires more diversity. Therefore, he has created grants for students in underrepresented backgrounds, allowing them to gain experience in positions of scientific research. Sello's motivation and love for research can be seen through his commitment and contributions in the field. MTB, or Mycobacterium tuberculosis, is a species of pathogenic bacteria responsible for causing tuberculosis, or TB. Tuberculosis is a serious infectious disease that mainly affects the lungs and is spread via coughs and sneezes. Symptoms of TB include coughing, weight loss, night sweats, and fever. MTB20S is a multimeric peptidase, meaning it is a protein with multiple peptide chains that is part of the proteasome in MTB. When MTB invades an organism, the host immune system releases nitric oxide in response to MTB. When MTB is exposed to nitric oxides, reactions occur between nitric oxide and/or its associative reactive nitrogen intermediates and proteins within MTB. These reactions are toxic as they damage MTB's proteins. MTB has shown resistance to the immune response, releasing nitric oxide because of 20S peptidase. A functioning 20S peptidase enables the bacterium to survive because the proteasome breaks down the degraded proteins that would otherwise impede cell function. MTB inhibitors inhibit or prevent 20S peptidase from functioning, which means that toxic proteins can't be broken down. Thus, MTB is no longer resistant against the reactions involving nitric oxide or its nitrogen intermediates. These are potential options for treatment, but research is necessary to determine if the proteasome inhibitors will inhibit human 20S peptidase, which we need for our own functioning. The potential of these inhibitors for treatment of TB inspired Sello's experiment. Selective inhibitors of MTB20S were designed and synthesized with the aim of finding inhibitors with high potency and selectivity. The selective inhibitors used were based on the specific substrate preferences and the structures of substrate mimicking covalent inhibitors of eukaryotic proteasomes called syringolin.
Here is a picture of the chemical structures of syringolin A, Sil A, and syringolin B, Sil B, proteasome inhibitors that were used in Sello's research. Sello needed to understand how bonding worked within Sil A and Sil B for his research. Chemical bonds are a form of attraction that connect atoms to each other and allow for the formation of various substances. There are three different types of bonds: ionic, metallic, and covalent. Ionic bonds are formed between a metal and nonmetal and involve the transfer of electrons between two atoms. Metallic bonds are found in metallic solids and involve delocalized electrons interacting with many nuclear centers. Covalent bonds are formed between two nonmetals and involve the sharing of electrons between two atoms. In both SIL A and SIL B, we can see that there are only covalent bonds that have formed between each element. For instance, hydrogen and oxygen located here are both nonmetals and thus have a covalent bond. Another way to determine if the bond is covalent is to use the differences in electronegativities of the elements involved in bonding. In this case, we would subtract 3.44, the electronegativity of oxygen, and 2.2, the electronegativity of hydrogen, to get 1.24, which indicates that the bond is polar covalent. The oxygen will have a partial negative charge because it has a high electronegativity, drawing the electrons closer to it and leaving the hydrogen with a partial positive charge. Electronegativity is the ability of an atom to attract electrons to its nucleus in a bond. Electronegativity increases across a period from left to right and decreases down a period from top to bottom on the periodic table. This is because as you move to the right of the periodic table, the amount of protons in the nucleus increases, increasing the effective nuclear charge or the attraction to the nucleus, so there is a higher electronegativity. As you move up the periodic table, less shielding occurs because there are less orbitals and energy levels. In other words, because there are more electrons in the atoms as you move down the periodic table, more shielding and repulsion occur, lowering effective nuclear charge. Thus, the atom will have less of an ability to attract electrons. We can see that the bond between the two carbons in SIL A is shorter than that in SIL B because double bonds are shorter than single bonds. We can also predict the molecular geometry of the compounds using VSEPR theory, or valence shell electron repulsion theory. Because electrons are negative and repel each other, we can predict the geometry since the electrons will be most stable when separated as much as possible from each other, or in other words, when a state of minimum repulsion is reached. To do so, we need to consider the number of domains, which are regions where electrons are found, and the number of lone pairs. For example, serengalin A and serengalin B have a methyl group at the end of the compound. Because the carbon contains 4 electron domains, 1, 2, 3, 4, we know that the methyl group has a tetrahedral electron group geometry. Since there are no lone pairs on carbon, it has the same molecular geometry as its electron group geometry, tetrahedral. Thus, there would be 109.5 degree bonds for the angles. Another example we can consider are the nitrogens in both serengalin A and serengalin B. Nitrogen has 4 domains as well, 1, 2, 3, 4, which means that it has a tetrahedral electron group geometry. But unlike the previous example, nitrogen has 1 lone pair. This means that the electrons in the lone pair will repel the other bonded atoms more than if there were no lone pairs, creating a bond angle that is less than 109.5 degrees. Therefore, the molecular geometry is trigonal pyramidal.
To conduct his experiment, Cello needed a good understanding of how solutions work. A solution is a homogeneous mixture of 2 or more substances. The substances are the solute and the solvent. A solute is defined as the material that dissolves, while a solvent is the material into which others are dissolved. One of the compounds tested during the experiment, compound 24, was synthesized by dissolving it in a 40% solution of TFA, or trifluoroacetic acid. In this case, the solute is compound 24, and the solvent is TFA. Molar concentration, or molarity, is the number of moles of solute per liter of solution. A 40% solution of TFA means that for every 100 liters of solution, there are 40 moles of trifluoroacetic acid. Internal chemistry determines solubility because of how ionic and molecular compounds behave. Specifically, ionic compounds, which consist of a crystal lattice structure, dissociate in water to form ions if they are soluble, or a solid precipitate if they are insoluble, while molecular compounds keep their overall molecular structure even if they are soluble in water, meaning they do not dissociate into individual ions. In this experiment, compound 24 is a molecular compound as it consists of nonmetals, which means that when it dissolves in the TFA solution, it will keep its overall molecular structure. It will not dissociate into individual ions. We can use stoichiometry and our knowledge of solutions to solve related problems. For example, consider the following scenario. 20 milliliters of a 0.3 molarity NaCl solution is prepared. What is the mass of Na+? Notice how we do not need to balance the chemical equation as it is already balanced. To solve this problem, we first convert the given volume of NaCl solution into liters because molarity is in units of liters of solutions. Then we multiply the volume by the molarity of the solution to get the moles of NaCl. Next, because there is 1 mole of Na plus in 1 mole of NaCl, we multiply the moles of NaCl by a 1-to-1 mole ratio to get the moles of Na plus. Finally, moles of Na plus is converted to grams by multiplying by the molar mass. From our calculations, we can determine that there are 0.138 grams of Na plus. If the problem involved a compound such as sucrose instead of sodium chloride, we would have to approach the problem differently. Sucrose is molecular, not ionic, so it would not dissociate into individual carbon, hydrogen, and oxygen ions in water. Thus, we would have to consider sucrose as a whole structure in stoichiometric calculations.
Overall, Seller's research found that the designed analogs weakly inhibit the human 20S proteasome and preferentially inhibit MTB20S over the human counterpart by as much as 74-fold. They can also penetrate the microbacterial cell envelope and render MTB susceptible to nitric oxide-mediated stress, which means MTB cannot infect the host. Furthermore, these inhibitors do not inhibit the growth of human cell lines in vitro. All of these results indicate that Sello's work is an important and promising starting point for tuberculosis drug development. And here are our citations.